Bismillahirrahmanirrahim. Assalamu alaikum dear students. I am your course instructor Idiva Muzammil, and today we are going to start the second lecture from the chapter Aging and Development. So our today's topic will be gastrulation. Gastrulation is the movement and arrangement of cells into germinal layers in the embryo. And the embryo at this stage is called gastrula. It is the second major phase of the embryonic development, which is characterized by the formation of three germ layers. Embryo in the gastrulation phase is transformed from a hollow ball of cells, the blastula, into three-layered stage called the gastrula. Three layers produced in gastrulation are embryonic tissues, which are called ectoderm, endoderm and mesoderm. And the mechanism of gastrulation varies depending upon the type of the species. Now the fate of the three embryonic germ layers. These three primary germ layers serves as the primitive tissues from which all the body organs will be formed. For example, ectoderm would change into the nervous system and skin epidermis. Endoderm forms the epithelial linings of the digestive, respiratory and urinogenital system and associated glands. And the mesoderm forms virtually everything else of the body. Now if we talk about the process of gastrulation, by the end of the second week of development, the bilaminar embryonic disc consisting of the hypoblast and epiblast has formed, which are shown here with the blue colored layers. Throughout the third week of the development, the bilaminar disc differentiated into the primary germ layers in a process known as gastrulation. And around 15 days after fertilization, this process of differentiation of the primary layers has been completed. The second structure forms along the midline in the epiblast near the caudal end of the bilaminar embryonic disc. Embryonic disc is a place where the two layers are running parallel to one another. One is shown with the blue color and the other one is shown with the yellow color, that is enclosing the yolk. This is called the primitive streak. At this stage, the formation of the primitive streak defines the major body axis of the embryo, including the cranial end towards the head and the caudal end towards the tail, as well as the left and the right sides of the embryo. The primitive streak expands at the cranial end of the embryo to create a primitive node in a circular depression, which is known as the primitive pit. This depression continues along the midline of the epiblast towards the caudal end of the streak. Caudal end is the tail end of the embryo, forming a primitive groove. Once the groove has formed, cells of the epiblast migrate inwards towards the streak, detached from the epiblast and slipped beneath it into the interior of the embryo. This process is known as invagination. The first cells to invaginate to the primitive groove invade the hypoblast and displace its cells. The hypoblast cells are now completely replaced by a new proximal cell layer, which is called the definitive endoderm. By day 16, the majority of the hypoblast has now been replaced. The remaining cells of the epiblast are now called ectoderm, which forms the most exterior distal layer. Some of the invaginated epiblast cells remain in the space between the ectoderm and newly formed definitive endoderm. These cells form a germ layer known as the mesoderm. Throughout the process of gastrulation, the ectoderm continues to form the cranial to the caudal end of the embryo, establishing three distinct primary germ layers throughout the whole embryonic disc. In the diagram, you can see the morula, which was a solid ball of the cells formed as the zygote undergoes the cleavage. In the second stage, you can see the early blastocyst, which forms a hollow ball of cells with a fluid-filled cavity. In the late blastocyst, the pre-embryo with the embryonic disc, two layers of the cells are formed that become the embryo proper. It contains the amniotic cavity filled with the amniotic fluid, the embryonic disc, the yolk sac, and different layers. The gastrula is the stage where the embryo with three primary germ layers, ecto, endo, and mesoderm, has formed. Next is organogenesis, and the first step of organogenesis is neurulation, which is considered as the first step of the organ formation or organogenesis. In this process of nervous system formation, as we know, epiblast turns into ecto and mesoderm, whereas the hypoblast forms the endoderm. And if we talk about ectoderm, it will form the nervous system. If you see in the diagram, the ectoderm becomes flat after thickening, and a structure is formed which is called neural plate. Neural plate is seen in 18 hours embryo of the chick, and after that the cells start to move inwards, forming a folding called neural groove. Neural groove becomes more prominent in 24 or 25 hours embryo. Afterwards, the neural groove joins to form a tube-like structure called the neural tube. On the upper side of the tube, the cells would form a layer called the neural crest, which forms the nerves of the sympathetic system later. Neural tube will be clearly seen in 29 hours embryo. The anterior part of the tube develops into brain and the rest of it forms the spinal cord. Now, today's assignment is what is spina bifida and what is the importance of folic acid to the growing fetus. Thank you so much.